Okay. Hey, this is another economic.
सरस्वत्यै नमो नित्यम् भद्रकाल्यै नमो नमः वेदवेदाङ्गवेदान्त विद्यास्थानेभ्य एव च मेधे सरस्वतिवरे भूतिवाभ्रवितामसि नियते त्वं प्रसीदेशे नारायणि नमोऽस्तु ते नियते त्वं प्रसीदेशे नारायणि नमोऽस्तु ते सरस्वती महाभागे विद्ये कमललोचने विश्वरूपे विशालाक्षी विद्यां देहि नमोऽस्तु ते विश्वरूपे विशालाक्षी विद्यां देहि नमोऽस्तु ते।
So, good afternoon, everybody. Am I audible?
Yes, sir. You are audible, sir.
Good afternoon, everybody. On behalf of organizing committee of Bharat Technology of today's webinar, I, Sandeep Kumar Pahari, welcome you all in this program. At first, I want to convey one message that this is the second webinar of our series of national webinar. The theme of today's webinar is good manufacturing practice and quality management system followed in pharmaceutical industry. But before going to start today's webinar, I want to say that our Bharat Technology started its journey since 2006, offering only B.Pharm degree. But now this college is offering 60 seats in D.Pharm, 100 seats in B.Pharm, and M.Pharm in some departments like pharmaceutical chemistry, industrial pharmacy, and cognosy. Now, I would like to invite you all for virtual overview of college. Richik Bandyopadhyay sir, please share the presentation. Sir, please. Hello. Thank you, Ritik sir, for sharing the presentation. Now, I must say that today Bharat technology is really lucky for the gracious presence of today's chief guest and speaker, Mr. Dishant Sanghvi, the Senior Manager, Global Audit and Compliance of Sun Pharmaceuticals. With it, today also we are thankful for the presence of our chairman, Professor Dr. Ravindra Devnath sir. Professor Devnath is not only a successful entrepreneur but also a pioneer in the area of pharmacy. He is the chief patron of today's program also. Now, I would like to invite him for his opening remarks for today's program. Sir, please, over to you, sir.
I think Chairman sir is not here still. You can continue.
Okay, sir, we can continue with our program. Sir, when will sir come? Then we will tell him to give some message.
Okay.
Right now, we will continue.
Later we can
Okay,
continue.
thank you.
Okay. Now I am taking the opportunity to introduce today's chief guest and speaker, Mr. Dishant Sanghvi. Mr. Sanghvi started his journey as an officer in Claris Life Science Limited in the year 2004. Then he discharged his duties as an executive from March 2005 to September 2009 in different pharmaceutical companies like Alembic Pharmaceuticals Limited, Zydus BSV Pharma Private Limited, etc. Then he was the senior reviewer in Apotec Research Private Limited for approximately 4 years. Afterwards, he was the senior manager of corporate quality assurance in Intas Pharmaceutical Limited. From December 2016 to till date, he is the senior manager of global corporate quality audit and compliance. He is ASQ, CQA, and PCS certified quality auditor and executed about 400 plus on-site audits worldwide, and he has 16 years of experience in different sections of different pharmaceutical industry. Now, without wasting his valuable time, may I request Mr. Dishant Sanghvi sir for enlightening us with today's topic, that is good manufacturing practice and quality management system followed in pharmaceutical industry. Over to you, sir.
Hello.
Hello.
Mr. Sandeep, your voice is not coming actually.
My voice is?
Yes. Yes, sir, it is audible.
Yes, yes, sir, audible. You are audible.
Hello.
Hello. You are audible, sir.
Hello.
Hello. Hello.
Sir, I think
Hello.
there is some problem in your earpiece. So,
Your voice is not coming.
sir, we can hear you, sir.
Probably he is not hearing us.
Okay, I make one call to him.
Hello.
Okay, sir. I am once again introducing you, sir. Please pardon. Now I am audible now, sir. Hello, sir. Hello. 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 Richik sir, am I audible?
Yes, sir, you are audible, sir.
Yes,
I
sir,
think
you
some
are
sort of
audible.
problem.
There is sort of problem in.
My voice is coming.
Yeah,
Yes,
yes,
sir.
Yes, sir, your voice
It
is coming, sir.
coming.
Your voice is coming.
Your voice is coming, sir. Hello.
I think sir left the meeting. There is some technical problem. So please wait sir, for a moment.
Hello.
Yes,
Hello.
sir.
Hello, sir.
Yes,
Now,
can you
now,
able to hear?
hello.
Can you able to hear me?
Yes, sir. Yes, sir. We
Yes.
are hearing you.
Actually,
And you are also hearing us now.
yes. Actually, your voice is not coming previously. Now it is okay. Now it is okay.
Okay, okay. I am once again introducing you, sir. Please pardon.
Yeah.
Now
Last.
I am taking
Yeah.
now I am taking the opportunity to introduce today's chief guest and speaker, Mr. Dishant Sangvi. Mr. Sangvi started his journey as an officer in Claris Life Science Limited in the year 2004. Then he discharged his duties as an executive from March 2005 to September 2009 in different pharmaceutical companies like Alembic Pharmaceuticals Limited, Zydus, BSV Pharma Private Limited, etc. Then he was the senior reviewer in Apotec Research Private Limited for approximately 4 years. Afterwards, he was the senior manager of corporate quality assurance in Intas Pharmaceutical Limited. From December 2016 to till date, he is the senior manager of global corporate quality audit and compliance. He is ASQ, CQA, and PCS certified quality auditor and executed about 400, more than 400 on-site audits worldwide, and he has 16 years of experience in different sections of different pharmaceutical industry. Now, without wasting his valuable time, may I request Mr. Dishant Sangvi sir for enlightening us with today's topic, that is good manufacturing practice and quality management system followed in pharmaceutical industry. Over to you, sir.
Yeah. Again, thanks to Sandeep Kumar for my introduction, and thanks to Mr. Vishnupada Biswal, Dr. Rabindranath Devnath, Dr. Biplav Devnath, Dr. Kunal Haja, Mr. S.K. Pahari, Dr. Sona Jana, Mr. Amral Vishal, and Mr. Bratati for giving me opportunity to speak at this national webinar, and all the students who have taken their time to attend this event in this Saturday afternoon. Thanks a lot. So just I will share my screen. I think host has to give me the rights for the sharing the screen.
Yes, sir.
Take me host. Yes. Now it is okay. Just a minute. Can you able to see?
Yes, sir. It is
Yes, sir.
visible.
Yes, sir, it is visible
Right.
and you are also audible.
Yes. Today's our topic is, as you know, GMP and quality management system in the pharmaceutical industry. So as you are the student of BPharm, then you will start with your job with the pharmaceutical industry, or you will start with some business with pharmaceutical industry. So I have prepared this presentation according to that. This will help you to assignment for your job as well as your business in the future. So we can start. As and when question, you can type into the chat box, and you can put don't to just put your answer in the chat box. So this is my request. Thank you. So we can start with first, what is good manufacturing practices. So if you go to any pharmaceutical industry, this word is you will heard many times till your joining, till your retirement. This GMP word is a very interesting subject for the pharmaceutical industry, good manufacturing practices. So what is good manufacturing practices? Every company must follow set of requirements to ensure their products are safe, effective, and suitable for the intended purpose. If we are taking the drug, means we are sick, and if sick people is taking the drug, there should be no any reaction. That disease should be nullified with the drug. That is the motto of any medicine. So for that, every pharmaceutical industry, starting from key starting material manufacturing, or API manufacturing, or drug product manufacturing, they have some sort of guidelines which they have to follow to adhere the requirement of the good manufacturing practices. So we will see one by one. So what is quality? So quality of drug is like a fitness for intent of purpose. So like if you are taking the medicine of the medicine for the fever, so that should be cure your fever. So it should be intended use of purpose. Then it should meet the conformance to the specification. What are the specification required for the respective drug? That should meet the particular specification. Then consistency in process performance and product uniformity. So it's
আপনার
like,
ন্যাশনাল হাইওয়ে এদিকে তো আমি যাব হাওড়া
yeah. So consistency in process performance, it is like, it is not like that end product should meet the requirement of the specification. Your process batch manufacturing should be consistent, meeting the customer needs. This is very important. As you manufacture any product, this should meet the customer needs, and a state from defects, deficiency, and significant variations. Like if you manufacture any product, if you are taking today X drug, and if you are taking the same drug after 2 years, so the efficacy of the product should be same. Then and then you can trust the respective product. So quality of pharmaceutical product is the ability to satisfy stated needs, including identity, strength, and purity without undesirable side effects. So there should be no any side effects. If there is an impurity in the drug product, then there will be a side effect. So it should meet their particular specification. So quality of the product is based on, like, it should be safe. So the product should be free from the unexpected side effects. Sometimes we take some drug and we have some reaction. All of us have faced this difficulty. So we can tell that is not a safe product. Identity. The product is exactly what is related to means the product should be there. The strength is the concentration of active ingredients. We are taking the product of 10 mg, 5 mg. So the strength what is described on the respective bottle or blister should be equivalent to that. The purity. The product is free from undesirable adulterating chemicals, biological and physical entities. So what are the ingredients written on the respective drug that should be there? No any other chemicals, biologicals or physical entities should be not there. And quality. The product meets standards and expectation of all customers. So if you maintain the quality, then all the customers taking your product is happy and they can cure their disease. So altogether, this is called SISPQ, SISPQ. So safety, identity, strength, purity, quality. So there are different regulation in the world. So if you have to market your product into respective country, then you should have their what are the procedure, and it is defined by their regulation. You should meet the particular requirement. Like if we can start with US, then if you go to US FDA, you must meet the ICH guideline. If you go to Health Canada, then you can meet the Health Canada HPFBI guideline. If Brazil, then you have to meet the NVISA guideline. Like that, if you have a different like UK MHRA, Europe EMEA guidelines, South Africa MCC. In India, from the CDSO, we have a Schedule M. Then if you go to Japan, we have PMDA guideline. If you go to Australia, TGA guideline. So as and when we export, then it should meet the requirement of the respective country. So all these countries having their set of guidelines. So that should be meet for particular medicine and their control. So to understand the GMP, we have to understand the six system of the entire pharmaceutical runs on the six system. If we are going to API industry, if we are going to drug product industry, altogether this six system is follow. So we will see one by one what are the system. One is a production system. Second is a facility and equipment system. Third is the laboratory control system. And four is the material system. Five is the packaging and labeling system. And altogether, we quality system. So any of our student or professor, have we heard this six system in the pharmaceutical industry? You can put your answer in the chat box. I will wait for 50-20 seconds and then I can go ahead with your answer and all. Yeah, somebody's tell yes. Yes, good. Good. So relationship among six system is the quality system and the five manufacturing system. The quality system provides the foundation for the manufacturing system that are linked and function within it. So like if you see this five system, production, facility equipment, laboratory, material and packaging, this all five system is controlled by the quality. And then we can tell we can have a good quality of the product. So we will see the system one by one, how the system works in the pharmaceutical industry currently. Just sorry. So first is a facility and equipment system. So facility and equipment system consists, if you see the picture, building and premises, equipment and instruments, utilities, and their commissioning, qualification, calibration, and maintenance of the equipment. This is considered comes under the facility and equipment system. So how we can facilitate design and how we can operation. So first we have to have a facility layout and man material movement. When we build any facility, we have to know that for which drug product we are manufacturing. If you go to the API manufacturing process is very tedious and a very lengthy process. There you don't have to consider mainly air and water. There is no any constraint parameter. But when you go to drug product facility, then there will be a specific environment for the drug product, and water and air quality should be as per the guideline. So there is a main difference between API and drug product, how you are manufacturing the product inside the. So clean room design and maintenance, then air handling unit design and operation, appropriate design airlocks. As and when you build a based on your product, you have to define your airlocks, like for gowning and de-gowning of the operators and the other personnel. Facility cleaning and their maintenance. So how you clean the facility and how you maintain to it, and appropriately the differential. To avoid cross contamination in the manufacturing facility, there is a pressure differential between the airlocks and the processing areas. So that will come into the next slide. And extraction system. Extraction system is like when there is some powder is altogether in the environment due to the manufacturing, that all powder will act by this extraction system and it will be thrown out in the environment. So if you see this diagram of solid dosage facility, like for tablet capsule, if you see the manufacturing area, we have like a coating room, solution preparation, encapsulation room, tablet room, compression room, and dispensing area. So in the solid manufacturing area, there should no any open product in the common areas. Altogether, we have to take into the respective processing areas. So as I told, there is a pressure differential and the respective processing area. So when is the negative pressure is there a clean air is coming from hallway into the room. So as and when we are taking the solid oral, means there are many powder generation is there. So powder generation is there means the powder should not from the processing areas, otherwise it will contaminate the other areas. So there will be a negative negative pressure. Altogether contradictory, if we are manufacturing injection, there will be a positive pressure. So particular contaminant from the hallway or corridor, it will not come into the respective processing area. Otherwise, because there is no any powder generation, the product is a liquid injection. So there should be a positive environment. So air gets contact with the product in the processing room. There will be a dust collector, which can be, if you see here, altogether dust is collecting in this dust collector, and through HEPA, it will go to the environment. So environment is also pollution free. All the dust will collect in this HEPA filter. So this type of the facility design will be there in the solid oral manufacturing areas. So if you build the facility, then it should be a segregated areas for the storage of rejected, recall, and return products, and quarantine status to be ensured. Why there should be a segregated areas? So if you are not put a segregated areas, sometimes what happen, you take the rejected product, take the batch. So there is a risk of that wrong product going into the market. So there should be a separate storage of areas, recall areas, and return product from the market. Then refreshment rooms and toilets separate from the manufacturing. Should not have a canteen facility inside the premises of the manufacturing facility. There should be a separate. Then interior surface like walls, floors, lining should be smooth, free from cracks, not shed particulate matter, permit easy and effective cleaning and disinfection. Why this? Because if it is there is a crack, there is a particular matter generation in the facility, it will directly go into the product and product will contaminate. So the area free from all these defects. Then repair and maintenance do not present any hazard to the quality of the product. And people, material and process must be unidirectional. What is unidirectional? Can anybody put in the chat box what is unidirectional? If anybody is knowing, I can wait for 30 seconds and then we can go ahead. So one answer is there in one direction. Okay. Okay, so you can tell this answer is correct, but I can give you the more information. Like unidirectional means the process which is starting from the warehouse till dispatch should be in a one direction. So altogether, the product will not come back to the manufacturing, and there is a no chances of any mix-ups. Otherwise, what happen if product is coming back to the other department, then there is a chance of the mix up and cross contamination. So the layout should be unidirectional. Then smoking, eating, drinking, and keeping personal medicine, cosmetic, and jewelry should not be permitted in the manufacturing areas, laboratories, production, and storage areas, etc. So this is a must requirement for the any manufacturing facility or R&D facilities. You should not do any of these things when you are doing the pharmaceutical manufacturing. Next most important part is the penicillin and cephalosporin manufacturing. These facilities are dedicated, and this will be a separate facilities or separate blocks in the pharmaceutical. Why? Because of this penicillin and cephalosporin, if the person is allergic to this penicillin or cephalosporin, it can be fatal to that person. So this facilities is quite different from the regular manufacturing of the pharmaceutical products. So equipment is also part of this facility system. Like if appropriate design and construction of equipment, if suitable size and suitable location, altogether it is there, then proper operation and intended use, effective cleaning, maintenance, and avoid cross contamination. So if we have appropriate design of construction, if we have a suitable size of equipment, and if we have a suitable location, then we can make a quality drug product. So this is a one picture I have taken to how the particular equipment, maintenance, and all things should be there. So if you are using any lubricant that should come in product, then it should be a food grade quality. And if any component like a pressure gauge, temperature gauge is joined to the respective reactor or respective equipment, it should be calibrated. Then there should be a labeling and drawing of the particular fixed pipe work. If you remember, there is all the pipeline is having a proper labeling and color coding. So if nitrogen pipeline is there, that is a blue. If it is a yellow pipeline, then it is a compressed air. So that the guideline is given in the respective SOPs and in the schedule M, and all the guidelines that what would be the color coding of the pipelines. And last is the contact surfaces. Surfaces of the equipment should not be reactive, additive, or absorptive SS316 grade, and thoroughly cleaned according to a fixed schedule. So why it should not be reactive, additive, absorptive? Because if you are taking any product, if the product, if the if the vessel is reactive to the product, then the vessel the product and it will contaminate the product. So it should not be the reactive, additive, and absorptive. Then equipment, there should be no any leaching of plastic components. There will be no any metal part which can be reacting to the respective product. There should be no any brittle gaskets. Lubricant should be the food grade and all, and should be no any paint chips and all. It should contact with the equipment. So if it is there, then it will be contaminating the drug. One of the most important part in facility and equipment is the cleaning. Why? Because if we clean the equipment and facility up to the mark, then and then we can ensure the product what we are manufacturing in the facility and the in equipment will be free from the contaminants. So for that, what we have to do? We have to follow approved cleaning procedure and using approved cleaning agent. It is not like that we can use any cleaning agent and we can clean the equipment or facility. There should be a approved cleaning agent. The determination of the efficacy of cleaning procedure used in production premises of an equipment. We have to determine like if there is a microbial growth in the facility and if we have to remove that microbial contaminants. So we have to determine like if X cleaning agent, if I am using, then it can kill the particular microorganism or not. Otherwise, the cleaning agent is not useful. Procedure will reduce the contamination to an acceptable level. So any of the contamination is an acceptable level, it is there. The cleaning procedure, what we are applying for the facility or for the equipment cleaning should reduce the contamination level. And the cleaning agent residue shall not lead to a cross contamination risk. Sometimes what happen, the what cleaning agent we are using remove the particular product from the equipment, but what cleaning agent is there, that residue will be deposit on the particular equipment. So that should be not there. Otherwise, that agent will be directly impacting on the next product. So that should we have to consider. So objective to demonstrate that after cleaning the production environment when return to original state of microbial and chemical cleanliness. So what equipment we have taken newly after cleaning, the equipment should be in the same state. So it should be chemically free from the last product and cleaning agent, and it should be microbiological also should be free. So one system we have done. Now we can go to the second system. That is a very much important system. It is a production system. So production system, we should have an adequate number of people who in working in the production system. Then people's qualification. So as and when in the pharmaceutical industry, we should have a relevant education, training, and experience. Like BCom or MCom student can't be part of the production system. If the microbiology is there, some microbiologists should be there to do the analysis of the respective test of the microbiology. Some other person with the engineering student or like that, they should not be qualified for the analysis. Then proper gowning procedure is there. If you go to the any industry like chemical industry, so chemical industry there is a lace gowning like overcoat and booties and all that. If you go to tablet capsule industry, there will be over gowning is there. Then there should be no any contamination from cubicle to another cubicle. But if you go to sterile facility, then your gowning, your booties, your hand gloves, your hood, and or your glass should be sterile. Why it is sterile? Because you are manufacturing sterile medicine product. So the contamination from the man to the drug, it should not. Then personal hygiene. So the person who is suffering from any disease or any open wounds are there should not allow to the manufacturing facility. Then cross contamination. Anybody knows the cross contamination? They can put on the chat box. Contamination and cross contamination. What is contamination and what is a cross contamination? I can wait for 1 minute and we can start. We can see the answer of the different person and we can start. Somebody choose mix up of real. Okay. I'm expecting for 2-3 answers more. If somebody can give, no problem. If it is a wrong answer, then no issue. You can give the answer. What you understand by contamination and cross contamination? Contamination means presence of impurities. Okay. Okay. So I will give the clear definition of contamination, cross contamination, and mix up. Altogether, first you have to understand all three are different. Contamination is a different material, different extraneous material like dust particle or your cloth particle or any particle will come into your product. That is contamination. Means that is not your part of your any other product. Cross contamination means if you are A and B two product, if A product go to B product or B product go to A product, that is called cross contamination. So that is a product to product. And if you are taking an example of mix up is a big kind of mix up. Like if you have a 5 container of X product and you have put sixth container of Y product for the dispensing of the product, then it is called a mix up. So these are the main terminology which you are using in the pharmaceutical industry. Then facility monitoring. Facility monitoring consider like a temperature, differential pressure, non-viable particle count monitoring, and viable particle count monitoring. And all together document should be there. Man, material and scrap flow, and here also facility cleaning. So how we can make the products of requisite quality? So there should be a right quality and quantity of materials, in process and sampling controls, and contamination control. If these three parameters is controlled very well, what right quantity and quality of materials, in process sampling and controls, and contamination controls, then we can manufacture products of the requisite label. This is the main part of any of the manufacturing process. Where should we label all materials? There in pharmaceutical facility, if you see any of or any of the glassware or anything without label, then it is a non-compliance. All the material, all time during processing, bulk containers, packaging lines, processing rooms, equipment should be labeled. If you are manufacturing any product into any of the processing areas, there also their status label is there, that this product is running into this particular processing area. And as far as manufacturing, cleanliness status also very much important. person can know this area is clean, this area is not clean, and labels should be clear, unambiguous, describe format. So one important parameter in the manufacturing facility is the line clearance. Why we have to take the line clearance? If anybody can give the answer. Why we have to take the line clearance in the pharmaceutical industry? I can wait for 30-40 seconds and we can go ahead. To avoid cross contamination, okay. That is correct answer. Anybody other can give answer? Okay, I will tell why line clearance is required. If you see this slide, work area and equipment are clean and free from previous product, previous starting material, previous batch labels, product residue, and last document. Why? So all this together will not come into the next product. So there should be no any mix up with the last batches to the this batch. So we can avoid the product recalls, market complaints of the line clearance. In the facility, line clearance either can be given by the production itself, production supervisor, or industry, it is given by the QA department, quality department. So if we go ahead to production system, there should be restricted access to production premises. It should not like that any visitor came into your production area because you are manufacturing the drug product. Process must be clearly defined and validated. SOPs, DMR, DPR, cleaning method and protocol. Correct starting material are accurately weighed, measured into clean and label container. Check all incoming materials to ensure the consignment corresponds to the order. So that is part of the warehouse. Each dispensed material, its weight or volume to be independently checked and recorded. There is one instance I have observed in one of my previous company where two excipients, these are the same name, sodium hydrogen phosphate and sodium phosphate something. So by taking a sodium phosphate beside of sodium hydrogen phosphate, they have manufactured the product, and when after filling it comes, the color of the product should be white, but it comes slight pale yellow. So they came to know the dispensing of the one of the excipient is wrong. So entire batch should be discarded because it is no use of that. Then approved material and those with their latest period to be used for the production. You can't use the expiry start material or packaging material into the production. Measuring, weighing, recording and control equipment instrument to be serviced and calibrated. The date of calibration and servicing, due date of recalibration to be indicated on a label attached to the instrument, and any spillage has to be reported immediately because it is a altogether chemical and all this. So spillage should not harm the personnel who are working in the facility. Correct. All the material and products should be stored under the appropriate defined condition. So as you aware, there is a different defined condition of the temperature condition of the material. Some is 228, some is less than 25, some is ambient condition. So all the material should be in the appropriate defined condition. Why? Because to have a particular material should meet the acceptance criteria as per their stability data. Checks on yield and reconciliation of quantity should be carried out to ensure that there are no discrepancies. Products should not be carried out simultaneously or consecutively in the same room. If in one room you have a sifter and you have a compression, then compression have a product and sifter is having a B product. That is not allowed in the GMP because it will cross contaminate the product. Any amendments to the manufacturing process, including any change in equipment or material, should be validated. And always verify time for company clock provided in the area where the activity is being performed. Don't use the personal watch. This is the one logical question. Why we should use company clock, not the personal watch? Give the answer. In the pharmaceutical industry, why we have to use the company clock, not the personal watch? I can wait for 30-40 seconds and we can go ahead if anybody can give the answer. Like if company clock is there, there are synchronized clock is coming into the pharmaceutical industry. So all this synchronized clock showing a single time in entire facility. If the production, if the QC, if the engineering, all together, there are same time in the facility. But what about our watch? Sometimes our watch is 10 minutes ahead. Sometimes our watch is 15 minutes behind. So we are making a note of a wrong time. So as a processing or we are analyzing any material into the pharmaceutical industry, we should log a time from the company clock. Yes, to avoid data integrity. Great. Then third system, we can go to this laboratory control system. So altogether, all the system having a document people and testing is a main part of the laboratory control system. Pharmaceutical raw materials, semi goods, finished goods, packaging material, reserve samples, stability testing, all are part of the respective quality control system. Then one more thing is analyst qualification in the laboratory. In the analyst qualification, what are the approved analysis there? Newcomer is coming, then newcomer and approved analyst, they are checking the comparison of both the analysts. Then and then a person can give the analysis of the respective instrument. Then analytical method validation is there. What are the method you are using should be validated. Then calibration should be there of respective instrument. What you are using, HPLC, polarimeter, UV visible spectrophotometer, all together, all should calibrated before the analysis. And out and out of trend result is also part of the investigation, is also part of the QC. So what is the principle of the laboratory system? All components are tested before use as per the test method. Result must conform to the specification for the release. And specification, standard, sampling plans, test procedure are established by appropriate quality unit. One most important thing in the pharmaceutical industry is a quality control should be independent from production. Here one question, why quality control should be independent from the production? If you can give the answer, anybody. Why QC should be independent from the production in pharmaceutical industry? If you go to any pharmaceutical industry in world, in global, QC is independent from the production. Independent means production person reporting to production head, QC to QC head, and there will be no exchange of the information in the production and QC. Any answer? I can give. There is a conflict of interest. If QC and production, suppose one person is working and one person in the QC, both are friends. So there will be a conflict of interest. What happen if the production, if they are not requirement of the respective drug after testing in the QC? So what friendly he can told you do the some setting and pass the product. So pass from the company, but when it go to the market, then it is not safe for the respective patient. So both should be independent, production and the QC. Just all finished product will be released only for sell only after the QC testing. So QC, what are the responsibility? They have to establish, validate and transfer and implement the QC procedures. They have to have a retention sample, oversee the control of retention sample. They have to do the stability study, so stability chamber management and stability study testing. They have to do the investigation of out of specification and out of investigation, out of trend investigation. They have to do the analyst qualification. They have to do calibration, and they have to do the timely release. Why timely release? If the products like what in the this corona situation, the remdesivir, the vaccine, altogether there are much in demand. So they have to do the time. It is not like that they will take too much time to release the product from the to the market. Then product specification and test methods. So they have to establish scientifically sound specification, sampling plans and test method. Material for packaging material, for in process, for finished product, for stability product also. So sampling is a one main part for the laboratory system. So approved sampling procedure should be have a method of sampling, sampling tools and equipment, how much sample to be taken for the particular product, subdivision of sample, because some sample they have to be tested chemically and some into the microbiology. So they have to part it into the two parts, one into chemical, one into microbial. Some sample container details. Container, if you are taking a microbial sample, then it is a sterile container. So sample container details is equally important. Application of container sample, special precautions and storage condition of the sample. So as and when you take the sample from 228 chamber, then when you are coming to QC, then you have to put that sample into 228. So you have to maintain the storage condition of the sample till the analysis. So testing. Testing should be validated. For compoundial method, verify the appropriate terms of the testing method before use. Any calculation should be critically examined. No repeat testing allowed. All failures incident to be reported and investigated as per SOP. And laboratory reagent solution, reference standards and culture media should be used based on available stability data. Next is a stability study. So what is the stability? Can anyone answer what is the stability study of the drug substance or drug product? I can wait for 1 minute. If you can give the answer, what is the stability study? If anybody can give the so I can give the answer. Stability is like a study. What your drug product is expired till how many periods? If you do the testing for 6 months, 12 months, 24 months, 36 months, so product is giving the same result as they are giving the result in the initial analysis. That is called the stability of the drug product or drug substance. So in what they are doing, initiate stability studies, selection of batches, how many batches they have to do the stability study, stability protocol they are doing, then sampling, receipt and chain of custody, sample and inventory and labeling, scheduling and withdrawal of sample, and sample testing, and data evaluation and trending. So one of the participants given the answer, stability supporting data that company used to establish product storage requirement. Correct. So good data and bad data. So what is good data and bad data? So has bench sheet of logbooks and that back it up, has acceptable preparation document, data is representative of the sample type, ugly data but not necessarily bad data. What are the data you are reporting in the QC analysis that should meet the all the requirements as per the good laboratory practice? What is the bad data? Only a final report, no supporting document. Sometimes what happen, analyst will not do the analysis. Just based on the previous batch, he will write the result. So that is not correct, because it will give the false information. No QC result traceable to the data, no load numbers. Sometimes they are not putting number on the worksheet. So we don't know that analysis result are of which batch, and final report result don't match the sheet result. Sometimes what happen, the certificate of analysis give the different result, and what are the inside sheets are there that is having a different result. So it creates the integrity of the result. Then fourth is a material system. What we can see, so material comes storage, material quarantine, facility, people and practices, hazardous material storage area, segregation of material as per requirement. Hazardous material should be segregated separately, documents, and distribution. This comes under the material system. So starting from material, raw material, excipient, packaging material, gases, solvent, process aids, reagent, labeling material, intermediate, bulk products, finished product, recall, return product, all comes under the warehouse facility. So material system start with the procurement of the material, then it will come to material storage facility, then the stock, and all together we can manage the particular entire material inventory. This is the general requirement of the material system. So no material used for the operations such as cleaning, lubrication of equipment, and should come into direct contact with the product. Where possible, such materials should be of suitable grade, food grade, to minimize health risk. Material and finished product should be quarantined immediately until they are released for the use of distribution. As and when some product coming to your pharma facility, then it should be quarantined. Store under the appropriate condition established by the manufacturer. If the manufacturer should be stored, then you have to put into the 2 to 8 chamber. If they are told that you have to keep away from the light, then you should not store the product under the light where the sunlight or this tube light is coming to the product. And stock rotation by FIFO and FAFO. This two terminology is very much important. FIFO is a first in, first out, and FAFO is a first expire, first out. So what are your first expire that you can take for the production? Otherwise, what happen? Your inventory management will be disturbed. So as we told, FIFO and FAFO, first expire, first out, expired rule should be there. Stock rotation should be there. Materials should be quarantined as and when it comes to the facility, and it should be stored under the respective storage condition. Starting materials should be purchased only from the approved vendor. If you are into the API industry, what are the key starting material is there, chemical is there, that should be come for the approved vendor. And if you are in the drug product facility, what are the API should be there, that should become with the approved vendor. And for this vendor, you have to have some technical and quality agreement. Without agreement, you should not buy the material from the approved vendor. All incoming material should be to ensure the consignment corresponds to the order. What are the order we have created and what are the material is came, that should be checked. Where additional labels are attached to the container info should not be the lost. It should not like that you can put the label on one on another. What are the previous labels should be there, that should be visible. And material supplied is free from the TSC BSC risk. So TSC BSC is like a kind of cattle. They are taking the making the material, so it will harm the respective mankind. So it should be free from the TSC BSC. If one delivery of material is made of different batches, each batch must be considered as a separate testing and release. If there are different lots are coming in entire one consignment, but the batches are different, then all the batches should be tested differently. Identity of the container of each container from which sample has been drawn. Only starting material released by QC/QA department and within their sample used. If QC and QA has not released the material, you can't use in the production. Correct starting material should be dispensed with accurate weight into clean and properly labeled containers. Each dispensed material and its weight or volume should be independently checked. If you go to any pharmaceutical industry, there are altogether in India, if you can tell, there are two procedures, done by and checked by. So one person is executor and second person is checking. So it avoid the mistake. Pharma. So packing material, the purchase, handling and control of primary and printed packaging materials should be as for starting material in secure condition, so as to execute the possibility of unauthorized access, store and transport in separate closed container. Outdated or obsolete primary packaging material or printed packaging material should be destroyed and its disposal recorded. Packaging should be checked on delivery by the packaging department for quality, identity and conformity. Absolutely no open material container in common areas or clean storage areas. Raw material to be dispensed in specifically designed areas, down for booths, and material containers to be cleaned before sent to warehouse. Rejected, rework and recall material. So if you see the picture, there is a cage. So in the pharmaceutical industry, rework or recall material is put into the lock and key, and sometimes lock and key with the warehouse manager, or it will the QA. So as and when material should come out, they have to take the permission from the quality department to avoid the unauthorized use of the material. Fifth system is the packaging and labeling system. So packaging and labeling system is having a packaging and labeling and artwork. Suppose you manufacture the product as per the good manufacturing practices, but if you put a mislabel, if you put a 100 mg per ml label or 25 mg per ml, then your product is correct, but your label is incorrect, then it will overdose the particular patient, because patient is prescribed 25 mg per ml and your label per ml, then the dose is not correct. So packaging labeling is also same important as far as the other system is concerned. So different products should not be packaged in a close proximity unless there is a physical segregation, otherwise it will mix up. Printed packaging material are critical to the conformity of the pharmaceutical product. Special attention should be paid for safe and secure storage of this material, and otherwise leads to the product recall. Otherwise, what happen if you have order of 25 mg per ml, and if there are certain bottles of 100 mg per ml, it came into the it go to the order, then customer will not accept the product, and you have to respective consignment. Here also line clearance is performed And for the packaging areas, to avoid the different previous product labels, foils and bottles, all should be removed before going for the next production. Packaging materials are tested prior. Components to be used must be checked and verified against the packaging order. The packaging department follow the packaging direction for each. This include affixing the lot number and the expiration date. So what are the batch number, expiration date, retest date to be checked before the dispatch of the product. So what you can check? Check and record the correct performance of any printing. Special care should be taken when cut labels are used and when overprinting is carried out offline. Roll feed labels are normally preferable to avoid mix-ups. What happens when you are using roll? So there will be no any other label will come into the respective roll. So there is a less chance of the mix-up. Online verification of all labels by automated electronic means to prevent mix-ups. Facility in the packaging area is having a camera system. Most of the good pharmaceutical industry, if you see. So what are the cases there? They have standardized with the image of that particular label. So other label it will come, then it will reject the particular bottle or particular. Challenge study to ensure that any electronic code readers, label, counters or similar devices are operating correctly. So regularly online control of the product during packaging should be included minimum checks on the general appearance of the packages, whether the package are complete, whether the correct products and packaging material used, whether any overprinting is correct, the correct functioning of the monitors. And sample taken away from the packaging line should not be returned. Otherwise, what happen? It will lead to the mix up with the other packaging components. Reconciliation amount of this is the main any manufacturing and packaging. If you have scrapped something, if you have taken a sample from the packaging, you all have to reconciliation. Otherwise, if you are not reconciliation, then what are the leftover quantities there that will be that can be unauthorized. Any unused batch coded packaging material should be destroyed and the destruction recorded. Checks to be performed before returning unused material if uncoded printed materials are returned to stock. Any failure of a batch to meet production specification should be thoroughly investigated, and the investigation should extend to other batches of the same product and another product associated with the specific discrepancy. So if some complaint comes with respect to some packaging, then you have to check the respective batch and your previous batches also. It is not like that you can check the only particular batch. Segregation of online rejection. As and when online rejection came, then you have to segregate. Otherwise, it will go to the approved products. Strict control over labeling operation. So labeling should be very strict. Otherwise, it will lead to the mislabeling. So this is the 5 system what we checked, and for this 5 systems, it is controlled by quality system. So we can see the quality management system now. This is the second part of the this presentation, QMS, quality management system. So what is a QMS? If anybody can tell, not full form, just a small definition. If anybody can tell, QMS. I can wait for 1 or 2 if anybody can give the answer. What is QMS? Sorry. Anybody from the audience? What is QMS, quality management system, in the pharmaceutical industry? Okay, I will tell. So QMS is the rules, behaviors, and ways of working in the pharmaceutical industry. How the quality is governed to the all other five system, that is the quality management system. If you can see the definition, a quality management system provides direction and control to the organization structure, procedure, process, and resource needed to the produce that is a fit for its purpose. So what is a QMS? QMS is a standard collection of policies, processes, document, procedures, and record and their associated responsibilities. In the pharma, what is there? If you have documented something, then and then you can tell you have done something. Without document, the executed activities will not taken into the consideration. So that should be a policies, that should be a procedures, and that should be a processes. So what is the benefit of QMS? Why QMS is required? If five system is there, it is not sufficient? No. Why? Because QMS will simplify and control the entire other five systems. So how it will simplify? So it will structure and return processes and procedures, opportunities to review and help the organization become more competitive, improve the communication within the organization, structure approaches in correcting defects, mistakes, or deviation. CAPA are consistently undertaken based on priority and risk, and CAPA process ensure higher risk problem and issues are promptly and dealt with, and improve complaint handling result to better customer satisfaction. So what quality system can do? It will simplify all other five systems to make a policies, process, procedure. So what are the complaint, what are the recall that are coming into the different department, it can be minimized. Clarify. QMS clarify roles and responsibilities. QMS helps understand the internal processes and how these processes link together. Employee understand where their contribution fits in the bigger picture. QMS will drive consistency in various processes as well as the continuous improvement. So what are the people, small people, like from starting from officer, executive, GM, AGM, what is their role to the pharmaceutical industry, to the main, their job? It will clarify by the QMS. It can control, what it can control? Control processes for better effectiveness and to increase efficiency, measure, monitor, and encourage continuous improvement in quality and productivity, which will become part of the organizational culture, and involvement of top management. So top management, how they will involve? Regular measurement, training, and reporting to executive management of critical indicators. If you see, if any regulatory industry come into the audit for the pharmaceutical industry, if they have seen any observation, will directly notify the owner of the company. They will not tell the QA head or QC head, inform, but as and when they will put the letter to the respective industry, they will put their owner name. Why? Because it is the top management responsibility that company should work as per the predefined SOPs. So by simplifying, by clarifying, controlling by the QMS, what will be the benefit? It will the better quality and product and services, lesser waste of resources, time will be lesser, and material waste should be not there. Improve customer satisfaction, and improve profitability, and improve bottom line. So that is the main motto of any business. So there are certain tools QMS are using. So that will come into next time. This is the two definition is given different guidelines. One is in ICH Q7. Each manufacturer should establish, document, and implement an effective system for managing quality that involves the active participation of management and appropriate manufacturing personnel. And it is advisable to extend the implementation of the QMS to the entire company. From the this perspective, all regulatory and GMP activities of a company are captured by the quality management system. So QMS is a requirement of the regulatory agencies. So there are certain tools for quality management system. There is an annual product quality review, change control, market complaint, personal qualification, kappa, that is corrective and preventive action, then self-inspection, deviation, incident, management review meeting, and quality risk management. So I have not taken the example of all the QMS tools. It will be a very big exercise, but I have taken a quality management tools example to have a understanding of the respective tools. So one example I have taken of the annual product quality review. If you go to any of the pharmaceutical company, any of the drug product or substance is having a annual product quality review. So what they are doing in entire year, what are the batch they are manufacturing, they will prepare a report. In the report, what they will take raw material and packaging material, what used for the manufacturing of the respective drug substance or drug product, that review they will take. They will check the product complaint. Is the product is having any complaint in the respective year? They will check the stability. They will check the in-process control and finished product trend and yield. They will record the reprocessing and reworking. Noted down any deviation, incident, change control, out of specification, out of trend, return goods, recall is there, and validation. Together, they will prepare a one report for one product. So it can give the entire picture of the respective product, how the product behaves in the respective year. Second, I have taken the example of the self-inspection. What is the self-inspection? So in the pharmaceutical industry, what happened? People are inspecting, one department is inspecting the other department. So what is the benefit of that? And why it is required? To measure the effectiveness of the quality system, provide objective evidence that adequate controls are in place, assure the product and process conforms to the specification, check adherence to process, ensure their compliance as per the GMP regulatory requirement, and ensure control, process and training systems are in place which ensure data integrity. So in pharmaceutical industry, all the industry having a audit system. And why in pharmaceutical is most important? Because it is gone, the product is gone to the patient. So they are doing the self-inspection to notify, is there any non-compliance, is there any department? And third example I have taken is the personal qualification. What we have discussed in the pharmaceutical industry, you should have a proper education, proper experience in the to join the certain department, proper skills you have, and proper training you have. Then you can working in the pharmaceutical industry. Only employees who are qualified shall execute their job responsibilities. Then when you join any department, you should know that what are the training you have to take, then you can start the work. Okay. So this is on my side. This is from my side, and I am open for the Q&A session. If any question from your side. Thank you.
Okay, thank you, sir. Is it audible right now?
Now you can take question, Amlan, if anybody.
Okay, sir. Thank you. Thank you for this beautiful message regarding the good manufacturing practice and also quality management system. Whatever we have learned about good manufacturing practice and QMS, which has been followed by ICH Q 7A guideline, I think, for quality management system. It is really very knowledgeable information you have provided to us. Some sort of questions were taken us from all our faculty members. So first question is, what should be the unidirectional procedure for sterile garments to provide it from autoclave to change room? What should be the unidirectional procedure for sterile garments to provide it from autoclave to change room?
sterile garment autoclave to change room. If you sterile garment, then the after sterile, the garment should be into the clean area, and from clean area it dynamic pass box. So dynamic pass box is having a HEPA filter on it, and from dynamic pass box it will come to the change room, and in change room also it will not put into like a open environment. It will put into the rack, where the rack is having a certain type of lights which can control the microbial contaminants.
Okay. Thank you, sir. Next question is that how cleaning procedure generally followed in batch to batch and product to product during manufacturing?
So this is, if I can take the example of tablet capsule manufacturing facility.
Yes, sir.
If there is a product to product, so what happens? Suppose I will take example of a product. So a product to a product. So they are any cleaning with respect to any wet cleaning. Wet cleaning means they are not using any water, they are not using any solvent, just they are using the equipment and they will remove the previous product powder altogether. But if they are going from A product to B product, means different product, then what they are doing, they are dismantling entire equipment. They will use the solvent, they will use the water, they will use the cleaning agent as per the procedure, and they will again put equipment all together into one, and they will take the line clearance and all like that, batch to batch, and product changeover cleaning place in the facility.
Okay, thank you, sir. So next question is that if a product which is already running in one manufacturing unit and the same product we want to run in some another unit, in different unit, then what sort of validation is required and what is that validation?
If you have a product in the facility and if you are taking the same product into the different facility, then first you have to prepare the equivalency report. This equivalency report consider the environment of the product, which is in I can say the A facility and B facility. So you have to compare all the things, equipment, products, personnel, all together you have to consider, and products, personnel, all together you have to consider, and based on the this equivalency report, you have to take the decision whether you have to have a require a validation or not. Sometimes what happen, A facility is having a stringent environment, stringent water quality and all, but B facility is not having that much type of automated equipments and all. There are manual system. Then if there is a difference between equipment, utilities and all, then to have validate the product in the other manufacturing facility also.
Okay, thank you, sir. A student from one a question from one student said, what is calibration, what is validation, and what is verification? If you please explain.
So calibration, verification, and third is
what is calibration, what is verification, and what is validation?
Your voice is breaking. Can you again? Calibration, verification,
and validation, sir. Validation.
Validation. Okay.
Yes.
So calibration is this terminology is for the components. Components like a temperature gauge, components like a pressure gauge, all together or instrument which are in the QC like HPLC, GC, balance. All together, if we are calibrating them with the like if I want to use a balance and if I want to calibrate, what I can take, I can take the calibrated weights which are created by the other agency, and I can put a weights into the balance and I can calibrate. So this is the calibration. Again, verifying this equipment that it is running as per the last calibration or not, that is a verification. So calibration and verification, it's all together, it is for the component and the QC instruments. if you are taking a example of validation, so validation is a different terminology which is if you are taking a process validation, then what are the consistency we have to take with respect to the products of different parameters, like what are the critical control parameters and critical control attributes for the respective products. If we have to maintain that, then it is a validation of the process. Type of validation: process validation, cleaning validation, transport validation, analytical method validation, analyst validation. So all together this comes under the validation. Means we have to have a data of consistency of this different type of activity.
Okay. Sir, can you please explain the flow of air through the laminar airflow in production unit?
Flow of air, it should be unidirectional. Like if there are two things are there. There are filters above and there will be a riser inside. So if air comes from the filter, then it goes to the riser. Like this, unidirectional airflow should be there. So and if some of the phase there is a no riser in down, up is the supply, and beside supply there will be a return. So what happened? Supply, the return will go to the from the roof itself. So as and when in a sterile facility, if I can tell what are the unidirectional airflow is coming during the in operation, when the people are working in that, then it should not what you can tell disturb the what are the activities people are doing. Otherwise it will contaminate the respective environment, and there should be no any dead pockets. Sometimes what happen, if there is a filter in one room, if there is a filter in one corner, but there is a filter in the second corner, so what happen, the air is coming into that area, but this area is dead. So there is a chances of the microbial contamination and other contamination.
Okay. Sir, a question from our faculty that what is data integrity? Is there any software followed in the industry to maintain the data integrity?
Data integrity, there are different guidelines coming to the data integrity. So if I can tell data integrity, there are two types. One is done by deliberately and one is not done by deliberately. If a person is like that, if I have to do something wrong to the product and I have done by deliberately, so this comes under data integrity. Means I have done and it is in my mind that I have to do that. Sometimes what happen, like if I can take the example, today is suppose today is the 1st of the January, but by mistake I have put the 1st January 2021, but I put the 1st January 2020 because the year changes yesterday, so I have made a mistake. So I have not done that deliberately, but it is altogether data integrity. And data integrity is a subject where the management should aware what people they are taking from the industry to the industry should come from very educational and a very skillful background. Otherwise what happen, any software or anything will not run the data integrity, because it is your soul and it is your what you can tell, it is your belief from how your grandfather and father, how they are make you till today, it will come into your job and your business. So there is no any software with respect to data integrity, but yes, there are many procedures come into the data integrity, and one recent is from PICS guideline came in this July month only. So you should refer that. And there is a very big topic, data integrity. So it can make one more session for the to discuss this topic.
Okay. Thank you, sir. Sir, another question is that what are the steps to be followed to prepare a zero defect product with consistency in manufacturing unit?
Can you again repeat?
what are the steps to be followed in manufacturing unit to prepare a zero defect product?
To prepare
A zero defect product.
Yes.
Zero defect.
See, if you can tell a zero defect product preparation should be very difficult, but if you have a first is a skillful manpower. Your operator and all things should be skillful. Your quality control system should be robust. Your what are the 5 system I told? What your facility should be unidirectional and free from the any contamination from outside. And your quality system should be very robust to run entire 5 system accurately. If you have all together, then you can make the product defect free. Otherwise, if you can tell only production can take a business to the pharmaceutical industry, then it is not possible. Altogether, this is a bunch of the different systems which can produce the defect-free products.
Okay, thank you, sir. A last question from us, sir. Is there any acceptable limit of after reconciliation of any batch?
Reconciliation, you are to the drug substance.
In drug product, sir, final product.
Yeah, final product. Final product, what happened? There are many things are there. First, there are samples. So what are the finished product samples are there? What are the steps are there? What are the rejections are there? And what are the control samples are there? That should meet the 100%. Otherwise, what happen if some vials or some blister, if it is not reconciled, then it will use in the next batch. It will lead to the mix-up. So 100% should be the criteria for the batch manufacturing in the drug product reconciliation.
Okay, sir. Another question right now came that is there any software used to in the to determine the QMS?
Yes,
Is
yes.
there any
There are
software
many,
available?
yes, yes, yes. There are many software people are using. Why? Because what happened now altogether all this thing will come into the digital. So and the facilities are too much big. So if I suppose if the manual system is there, if I record any change control, I have to walk and I have go to the warehouse, like that I have to do. But what happened in the computer itself, there is a software called TrackWise. There is a software called DCS, Document Control System. These are the softwares which are available in the market, and you can customize that software also as per the procedure of the respective industry, which can be used for the change control, deviation, incident, CAPA management, internal audit, external audit. So it can minimize and it can monitor activity automatically. Otherwise, what happen? You can do the backdating, forward dating, all this thing. But if you computer, if it is there, then you can have the respective data from the computer itself. You can't do the data integrity from the software.
The last question is that if a product has been found default in market after going to market, if we found any defect in the product, then how we generally do the recall? What is the recall procedure? If you can.
There are different things applied into the regulatory and domestic production. If regulatory, I can tell there are two kind of things. One is the voluntary recall and one is recall done by the FDA inspector. So what happened? The customer will notify the respective industry that this is the complaint we have received. This is the defect. Investigate, and if the respective defect is a part of that particular company's issue, what they will do, they will voluntarily recall the entire products from different countries what they have given into the market. And if they are not doing, FDA will emphasize to do the recall. If you are considering the domestic market like India market, there is a very less recall and it's done. Why? Because people are not so much what you can tell, they are educated but not so skilled where to complain, how to complain, if complaint is done, then how it will trigger to the respective company. So that mechanism in the domestic market is still not as far as we are comparing with the regulatory market.
As a last question from myself only, and what generally we used to do with those batches after taking the validation batch, 3 validation batch, what generally we used to do with those tablets after the validation batch, is after performing the validation batches, what we do with those tablets? Shall we taken those tablets to the market, or we keep it in the industry only for further
See,
checking of
there are
parameters?
three kind of validation. Now is a prospective validation, concurrent validation, continuity verification. So mainly people, what they are doing, they are taking a validation on the small scale. It's not like a bigger size, batch size they will take. Why they are taking a validation? They will take that particular batch and put into the step. Yeah, sorry. So they will put and if they are taking after 3 batch, 4th batch taking a on large scale. Sometimes what happen, there is a like if you see this situation of corona, so people are manufacturing remdesivir, so there will, they can't take the exhibit batch, they can't do like that. So what they are doing concurrent validation. So entire batch they will commercialize and the same batch they will put for the stability.
Okay. Thank you, sir. Sir, after all, it is a very good session. We have enjoyed fully this session. And last, a request from all of us is that please, sir, give us some information that how a freshers candidate should prepare himself for getting entry into the industry. If you please give us some information.
Hello? Hello, can you hear me? Hello, can you hear me? Hello?
Yes, sir. Yes, sir. It is audible, sir.
Yeah. Can you hear me?
Yes, sir. It is audible.
Yeah, it is audible. Good afternoon, all of you.
Yeah, good afternoon.
Okay. Good afternoon, sir. Good afternoon.
So our invited speaker from the really is wonderful. With other methodological day by day syllabus curriculum also developed so that our main motto to supply quality medicine to the patient and rational medicine to the patient. So only if the students are study the materials thoroughly and they also, first of all, the quality is the first, then material is the quality product is, and now industrial development in India is very vast. I mean, quality medicine as well as products, everything is available in India. Now, in COVID situation also, India is parallel to other developed countries. That is our scientists, they are working a lot. They are developing newer vaccine or medicines also will come out. And once the COVID will be totally controlled by our scientists. So these are the main aspects. So from industrial development, industrial subject is to be studied. Our diploma pharmacy course is totally now revised on on clinical and hospital pharmacy base, clinical hospital pharmacy community base, and our syllabus for B.Pharm and M.Pharm, a part of B.Pharm and M.Pharm is also from industrial and research base. So the days are coming when India will be a number one in the country for producing quality medicine and quality manpower for our country personnel, for our patient, for our mankind. So it is separated from our petrochemical department. So pharmacy is developing in in the all aspect on the quality medicine, patient counseling, cultivation collection, industrial development, and thorough analysis, good type of analysis, so that no impure or adulterated drug can be marketed. Our academician as well as administrator, they are very much honest in this field. So they will go, this profession will go a long way for quality medicine and quality supply of the patients who are suffering from various illness. Thank you very much. Many thanks. You have participated to our.
Thank you, sir. Thank you for your speech. And okay, so continue with our program. I'm asking a question to our respected delegate speaker, sir. Sir, our last question from all of us, that if you kindly give any sort of information to the freshers candidate, those who are trying to join in the industry, what sort of preparation they used to take to gain more knowledge, to gather more knowledge? If you please explain.
Yes. So main things, if you can consider the India pharmacy colleges and all, there are many colleges are running now in different states, and other states are open for all for job and all. And if you can consider like Gujarat, Maharashtra, and if you go to north side and south side like Telangana, Andhra, altogether they have many industry for the pharmaceutical. So first of all, you have to decide that which state or which region is preferable to you based on your nature and based on your skills, inherent skills. And altogether, all the CVs are look the same. Like particular person has done the B.Pharm or M.Pharm, the respective college. But if your skills, you can improve your skills by reading the guidelines from the industries, regulatory authorities, or by taking the trainings from the various speakers from the different companies, or by taking some courses which are available on the websites, or which are might be sometime it is or sometime it is paid courses. So what happen, your CV will different from the other CV. Otherwise what happen, the CV of all the candidates looks the same. So if you make your CV different from the others, you should have some extra certification, some extra knowledge you have, then you can survive in the industry. And nowadays, it is very difficult to enter into the pharmaceutical industries. So better to have a different skills, you can emphasize on your development.
Okay. Thank you, sir. Thank you for your message towards our recent freshers candidate who are willing to come to the industries. Now I will pass my stage to Mr. Sandeep Pahadi, sir, for further proceedings. Over to you, sir. Sandeep, sir. Sandeep Pahadi, sir, is it audible?
Okay. Now
Yes, sir.
it is audible now,
Yes, sir, it is audible. It is audible.
but video is not being started. It should be controlled by your yeah, committee members.
Okay.
Okay, anyway, thank you, Amlan sir and Mr. Dishant Sangvi sir. I just like to request our Sangvi sir to stay few minutes with us. Our principal sir, Professor Dr. Kuntol Hajra, wants to share his remarks for today's function. I am requesting our principal sir to share his remarks. Sir, please.
First of all, I really want to thanks Mr. Dishan Sangbi for his valuable time with us. We got a very informative. He has given a very informative. It was very informative session. We gain a knowledge about GMP and quality management system. And I observed that our question answer it was so much lengthy because every year cleared our faculties as well as our students doubt. It was really an interactive session and really a knowledgeable session, sir. Once again, I want to thank you, sir. Thank you for enlightening us. Thank you for your valuable time. I can expect that in the future also, sir, you'll help you'll extend your hand
Definitely.
towards us. Thank you, sir. Thank you, sir.
Thank you, Kuntal sir. Now the organizing committee is taking the opportunity to felicitate our respected Dishant Sangvi sir virtually by an e-memento and e-certificate. To do this, I request Mr. Richik Bandyopadhyay to felicitate him. Hello.
Hello.
Hello. Hello.
Yes, sir.
Hello.
Yes, sir.
Once again, I am requesting our Richik Bandyopadhyay sir to felicitate our distinguished guest, that is today's chief guest and speaker, Mr. Dishant Sangvi sir, virtually by an e-memento and e-certificate. Please felicitate him. Hello.
Is it visible, sir? Is it visible?
Yes, it is visible, sir.
Yeah. Thank you,
And-
sir. Okay, Amlan sir, you can continue.
Yes.
actually.
Thank you, sir. It is the certificate from all of us, from our BT family, Bharat Technology family. We are providing a certificate of appreciation to you for this today's session. And also, along with this certificate, we are providing you a memento. This is our memento from our side. So this is the memento given to you, sir. And we will also mail this memento and also the certificate in your mail ID. So virtually, we are showing this memento in this platform. Please accept this, sir.
Okay, thank you. Thank you very much.
Thank you, sir. Thank you. Thank you.
Welcome, sir. Welcome, sir. Hey, I am audible.
Yes, sir. It is audible.
Welcome, sir. Now we have come to an end of today's webinar. For offering vote of thanks, I would like to invite Mr. Vishnupada Biswal, Assistant.
Hello.
Thank you, Mr. Sandeep Kumar Pahadi, sir. So, good afternoon, one and all. It's my privilege to have been asked the vote of thanks. So, first of all, I would extend my sincere thanks and wholehearted gratitude to our today's eminent speaker of today's webinar, Mr. Dishant Sanghvi sir, for accepting our invitation. First of all, in spite of his busy schedule of time, and moreover, I would like to request him, sir, please do continue support like this in future also whenever we need you. I would like to thank him also for his such a wonderful, informative, and also so nice elaborative session in today's webinar. Once again, thank you, sir.
Yeah,
From the core of my heart, thank you, sir. So, from the core of my heart, I would like to thank our chairman of the institute, today's chief patron of today's webinar, Professor Dr. Ravindra Devnath sir, for his continuous support for conducting such type of webinars. And also, I would like to thank convener of today's webinar, Dr. Biplab Devnath sir, and principal of the institute, come chairman of the today's webinar, Professor Dr. Kuntal Hazra sir, for organizing such a wonderful webinar. And my heartful thanks to coordinator, moderator, and also all technical staff and my beloved colleagues and also non-teaching staff who helped a lot. I think without them, this type of webinar would not have been a successful one. Last but not the least, I would like to thank my sincere thanks to all the participants who have participated in today's webinar and also who made this webinar a grand success one. So once again, thank you one and all. Thank you, sir.
Yeah, thank you. Thank you from my side also. Yeah, I can tell it will surely help for the student for their future assignment wherever he goes, like for job or business. Thank you from my side.
Thank you, sir.
Thank you,
Thank you.
sir.
Okay. Thank you, sir.
Thank you.
Okay. Okay. Can we take leave?
Okay,
Yes,
sir.
sir.
We will end our session here
Yes,
only.
please.
Thank you, sir. Okay.
Thank you.
Thank you. And
Bye.
we invite you once more in your future.